Hello, everyone, and thank you for joining us today for this LEXOGEN webinar organized with Iowa State University. My name is Lauren, and I'm a U.S. application scientist at LEXOGEN. With me is Josh Franken, LEXOGEN Central U.S. Sales Manager, and Michael Baker, who is the facility manager at the Iowa State University DNA facility. Today, I will be discussing two of LEXOGEN's NGS library prep products, QuantSeq and QuantSeq-Pool, which allow for ultra-plexing for the highest efficiency and lowest cost differential gene expression studies.
I want to thank you all for attending this webinar and your interest in digital gene expression. I just want to quickly mention what the Iowa State University DNA facility has to offer with respect to digital gene expression. As you will see today, digital gene expression is a much more cost-effective method compared to standard RNA-Seq for differential gene expression studies. The Iowa State University DNA facility's primary digital gene expression library prep is LEXOGEN's QuantSeq 3' mRNA-Seq forward library prep. The current price for our QuantSeq prep is $23 to $30 per sample. Compared to our standard RNA-Seq prep at $112 to $154 per sample, this represents a cost savings of about 500%. Additionally, since only 50 to 100 bases of each transcript is sequenced, the researcher only needs about 5 million reads per sample. With standard RNA-Seq, the same experiment would require about 10 times more sequence data per sample. Therefore, not only will the researcher be realizing cost savings with the library prep, but now more samples can be indexed per lane, considerably reducing the sequencing necessary for a project. Our facility also offers LEXOGEN's Globin block, which removes Globin transcripts from blood samples before library prep. We use Globin block in conjunction with our QuantSeq service, which adds an additional $4 to $7 to each library prep. We have primarily used the pig kit, but LEXOGEN has Globin block for other organisms as well, and has worked with researchers at Iowa State University to develop a custom Globin block for a turtle species. For additional information regarding QuantSeq sample requirements and submission, please contact Tanya Murtha or myself. If you are interested in any of our other services, please check out our website at www.dna.iastate.edu. If you don't find what you are looking for, please contact us. We are certainly willing to work with you. With that, I will turn it back over to Lauren.
Great, thank you so much, Michael. So now I will introduce you to LEXOGEN. So LEXOGEN is a biotech company focused on enabling analysis of the complete transcriptome. LEXOGEN was founded in 2007 and is based at the Vienna Biocenter in Vienna, Austria. We also have a subsidiary in New Hampshire, USA, founded in 2014. Our company is comprised of a multinational team and a worldwide distributor network, allowing researchers across the globe access to our products and services. Our overall mission at LEXOGEN is to allow researchers to exploit the complexity of the transcriptome to drive all areas of research. LEXOGEN offers an extensive portfolio of products to enable sequencing of the transcriptome. Although this webinar pertains specifically to library preparation products, I do want our listeners to be aware of the end-to-end workflows we have available. So shown here is SLAM-seq. SLAM-seq is a method for the measurement of newly synthesized and existing RNA in cultured cells. It enables resolution of RNA synthesis and degradation kinetics. Our SLAM-seq portfolio includes Explorer and Kinetic modules, which covers the entire metabolic RNA labeling experiment, from optimizing labeling conditions to labeling nascent or existing RNA and alkylation for downstream NGS library preparation. Total RNA from SLAM-seq experiments can be used directly with QuantSeq, which I'll discuss later in this presentation. We also have a data analysis pipeline for analyzing SLAM-seq data called SLAM-Dunk, which is available on BlueBee Genomics platform. As far as the bottom of the graph, I will cover this in a couple of slides. Here you will see our available products for preparing your RNA, as well as generating libraries for sequencing. For RNA preparation, we have Split for extraction of RNA from a variety of sample types, now including SARS-CoV-2. Trapper is our small RNA isolation kit, which isolates all risk-associated small RNAs found in any organism, tissue, cell type, or biofluid. For ribosomal depletion, we have RiboCop for use in human, mouse, rat, or in bacteria. And our poly-A kit is for the rapid and highly specific enrichment of polyadenylated RNAs from total RNA samples. For NGS library preparation, we have 4 main kits depending on the needs and goals of our researchers. QuantSeq is our gene expression analysis kit, and again, I will go into this product in more detail in the upcoming slides. For whole transcriptome sequencing, we have both Coral as well as Sense. And our small RNA-Seq kit allows for the generation of small RNA libraries for Illumina sequencing directly from total RNA or enriched small RNA. Our full-length cDNA preparation product is Teloprime, and we also offer spike-in RNA controls called SERVs. For more information on any of these products, please visit our website. Finally, we have several pipelines for NGS data analysis. On our BlueBee Genomics platform, we have pipelines for analyzing data from QuantSeq, Coral, and SlamSeq experiments. And these data analysis tools are simplified, allowing users without bioinformatic knowledge to utilize these tools for data processing. On the bottom of this flowchart, you will see that Lexogen also offers in-house services for the preparation, sequencing, and data analysis of your samples. Again, please visit our website or feel free to reach out to us for more information on any of these products or services that I've mentioned. So I now want to transition over to discussing the 2 major types of RNA-Seq. So up until now, whole transcriptome sequencing, be it mRNA or total RNA-Seq, is what has been used to look at de novo assembly of transcripts, detect isoforms, and perform expression profiling. With this technique, you have multiple fragments generated per transcript and end with a fairly involved bioinformatic analysis, which results in a lot of time spent and high cost to obtain your NGS sequencing results. For expression profiling, this is much easier and straightforward approach because you build your library around the fragment of interest and then simply sequence 1 fragment per transcript. Because of this, you end with a much simpler bioinformatic analysis, essentially counting the number of sequences and eliminating assembly and normalization. Overall, with this expression profiling method, you can save 1/10 of your sequencing reads because there is no need for mapping the entire transcript. Because of this, you're able to multiplex higher and spend less on computational resources due to the simplicity of downstream data analysis. So this table is showing more comparison between conventional and 3' mRNA sequencing. For sample types, high-quality RNA is typically required for conventional RNA-Seq, whereas with expression profiling, low quality, including FFPE samples, can be processed as well as high-quality samples. Time differences between the 2 methods are significant, with 3' mRNA-Seq not requiring fragmentation. I won't discuss all these items on the table as I've already touched on them in my previous slide, but I hope you can see that there are major advantages that result in lower cost overall when choosing 3' mRNA-Seq. So this brings me to Lexogen's solution for expression profiling, QuantSeq. So QuantSeq started as a single product, which I will first cover, but it eventually branched out into a family of products, covering a variety of research goals. In addition to QuantSeq, there is QuantSeq Flex, which allows for targeted sequencing. I won't be discussing this product today, so if you have any questions, please feel free to reach out. QuantSeq-Pool is our most recent product release, and I will be discussing this later in the presentation. So for QuantSeq and QuantSeq-Pool, these kits use the same methodology of binding to the 3' poly-A tail of the transcript for initiation of reverse transcription. In QuantSeq Flex, the researcher has the option of designing their own oligos for binding at a specific location in a transcript of interest. So I will now go into detail on our QuantSeq 3' kit. So as previously mentioned, with this approach, you can utilize either high or low quality RNA, including FFPE samples. The oligo for first strand synthesis targets and binds to the 3' poly-A tail, and reverse transcription proceeds. Following this, RNA is removed and a random second strand synthesis is carried out to produce double-stranded cDNA libraries. In both first and second strand synthesis, partial Illumina adapters are included in the 5' ends of each oligo. Library generation is concluded with a purification step to remove any small artifacts and primers. The library is then amplified by PCR, and complete adapters are incorporated for clustering on a flow cell. Additionally, 6 nucleotide indices, either I7 only or I7 and I5, are introduced. I will get into the different indexing options later in this presentation. After library amplification and purification, your libraries are ready for quality control and subsequent sequencing. For sequencing, read 1 will generate reads toward the 3' poly A tail, so your reads directly reflect the mRNA sequence itself. For QuantSeq, we don't recommend paired end sequencing because read 2 would be very low quality due to the poly T stretch at the beginning of read 2. So far, I've shown you the workflow for QuantSeq Forward. However, I do want the audience to know that we do have another product called QuantSeq Reverse. So with QuantSeq Reverse, it is possible to pinpoint the exact 3' end in read 1. Hence, this product is used to study the 3' UTR and alternative polyadenylation sites. For QuantSeq Reverse, we do advise customers to perform paired end sequencing, and our kit does include a custom sequencing primer, or CSP, that must be used in sequencing. So next I will get into the performance of our kit. As mentioned before, QuantSeq libraries cover the 3' end of transcripts, so you have significant savings in terms of read depth. In the graph to the left, you will see a major benefit of QuantSeq when plotting the relative coverage across the normalized transcript length. Standard mRNA-Seq distributes reads across the entire length of transcripts with underrepresentation of the 3' and 5' ends, which you can see with the gray line and gray shaded area. Whereas QuantSeq covers the very 3' end of transcripts. And as you can see, it's the blue line and green shaded area. For gene expression and differential expression analyses, 1 read per transcript is sufficient. So the additional sequencing space gained by focusing on the 3' end can be used for a higher degree of multiplexing. In the present example, standard mRNA-Seq has a 12.4-fold higher relative sequence coverage, which in turn presents the maximal possible reduction in read length when using QuantSeq while still determining gene expression accurately. When comparing mapped reads between QuantSeq and conventional RNA-Seq, focusing on differential expression metrics, you'll see that these results are very similar. You'll notice one outstanding feature unique to QuantSeq, in which reads map to no feature to a higher degree compared to mRNA-Seq. This is largely due to incomplete gene annotation at the 3' end. Reads are mapped correctly to these unannotated 3' UTRs and poly A sites, but are not counted in the protein coding category due to lack of annotation. So correction of an incomplete annotation or a sensible 3' extension of the gene definition can address these discrepancies between the available annotation and the real sequence transcriptome. Overall, QuantSeq provides you with reliable data to perform differential gene expression studies. So I mentioned previously that QuantSeq can be utilized with low quality and FFPE samples. This is a very nice experiment illustrating this. So xenograph of a human tumor cell line was split into 2 pieces and processed as a fresh frozen cryo block with a RIN of 8.3 or embedded FFPE material with a RIN of 2.8. QuantSeq libraries were generated using 50 nanograms and sequenced on HiSeq. As you can see in the plot to the left, there is a high correlation in gene expression between these 2 libraries. Regarding gene detection, the detection threshold set at 5 reads per gene, the difference in gene detection was only 3%. The high concordance of gene expression detected in cryo and FFPE RNA samples demonstrates that QuantSeq is the method of choice for gene expression quantification independent of the RNA input quality. So this is all the data that I will be showing for QuantSeq today, but if you are interested in obtaining more data or publications on QuantSeq, please feel free to reach out to us. So as mentioned earlier, we also have a pipeline for the analysis of QuantSeq data, and this is hosted on BlueBee Genomics platform. I wanted to touch on this because if you do order a kit from us, you will see that we provide an activation code for the number of reactions in the kit, which you can then use for data analysis. This platform is extremely user-friendly, and again, no bioinformatic knowledge is required. The environment is extremely secure and allows you with multiple options for uploading and downloading your data. We have many species available, But if you would like an additional one added, we can do this for a fee. Just feel free to contact us. And finally, we have a promotion going on until the end of this year, where if you need additional codes for processing data, you can obtain these for free. So now I will touch on a few modules that can be added onto the QuantSeq kit depending on the researcher's needs and goals. So this module, called Globin block, is specifically for blood samples and results in the blockage of library fragments generated from the abundant and highly stable Globin mRNAs that are present in whole blood. The Globin block modules consist of a modified RNA removal solution, and in this kit it's called RS Globin block, which contains species-specific oligos complementary to highly abundant Globin mRNAs. Unlike other currently available Globin depletion methods, which remove Globin mRNAs from whole blood RNA prior to library preparation, this module enables Globin reduction during library preparation itself, making it very cost-efficient and easy to utilize. Also, current approaches require higher input amounts of total RNA, typically over 1 microgram, and add additional hands-on time for pre-processing of RNA prior to library preparation. So with Globin block, you can remove Globin mRNAs from down to 5 nanograms of total RNA. So right now we currently have 2 modules available for human and pig samples. QuantSeq kits provide you with I7 indices, but depending on your multiplexing strategy, you can add I5 barcodes for up to 9,216 combinations. Benefits of this include the ability to detect index hopping and minimize misassignment of indices. We also have an online balance checker tool depicted to your right, so you can choose the indices that are well balanced. We also have our 12 nucleotide unique dual indexing, or UDI option available as well, which allows you to have 384 truly unique dual indices. The benefits of including UDIs are for their error correction capabilities, which is a result of index sequencing error, for which we have a free tool available if you do use this product. Our UDIs are easy to use and scalable depending on your needs. In addition to being an add-on option for QuantSeq, we also have QuantSeq kits available with UDIs incorporated. And we also have options available for Coral kits, which is our whole transcriptome sequencing library prep kit. Additionally, UDIs are compatible with non-Lexogen libraries as well. The final module that can be used with QuantSeq is our unique molecular identifiers, or UMIs. These tags allow you to detect and eliminate amplification bias. So similar to Globin block module, it's a simple reagent swap, so it's very easy to incorporate. Additionally, we also have QuantSeq kits with UMIs included, available for your purchase. And similarly to our UDIs, we have a free command line analysis tool for the deduplication of your UMI tagged transcripts. This concludes the presentation on QuantSeq. Overall, I hope I was able to show you how cost-efficient expression profiling is. And with QuantSeq, you are able to multiplex a large number of samples due to the sequencing space saved by using this method. One thing I didn't touch on is the low inputs that can be used with QuantSeq. So researchers can input as little as 100 picograms of total RNA. We do have modifications to our protocols that can be used when using such low inputs, as well as low quality or FFPE samples. And as I mentioned earlier, QuantSeq is also available as a flex option, allowing researchers to design and target specific transcripts. And finally, our data analysis pipeline for QuantSeq is simple to use and free with the purchase of a kit. If you have any outstanding questions regarding QuantSeq, please feel free to type them into the chat window or email us at support@lexogen.com, and we will be happy to answer them. So now we'll move on to talking about our newest product, QuantSeq-Pool. So similar to QuantSeq, QuantSeq-Pool targets the poly A tail of the 3' end of transcripts for initiation of reverse transcription. The major difference, which I will get into, is the ability of early pooling of your samples into one reaction. As you can see, the workflow and methodology is very similar to that of QuantSeq. So I will now point out some of the differences. In the oligo DT primer used for first strand synthesis, there is a sample barcode called I1, and also UMIs included. Following reverse transcription, samples are pooled into one reaction, allowing for streamlined processing. This eliminates the need for QCing individual libraries, saving researchers significant time. Indexing is optional due to the I1 sample barcode included in first strand synthesis and will ultimately depend on the number of samples to be multiplexed. Overall, the most important takeaway from this is that QuantSeq-Pool allows you to pool up to 96 samples into one tube. Because this pooling is done early in the protocol, it allows for a very simplified workflow, helping to keep things consistent and efficient. QuantSeq-Pool enables robust and consistent gene detection even at low sequencing depth, as shown here. For this experiment, QuantSeq-Pool libraries were generated from 10 nanograms of high-quality RNA per reaction, sequenced, and gene detection was analyzed at 100,000, 500,000, or 1 million reads per sample. The number of detected genes was counted at a threshold of over 10 counts per million, or CPM, which shows highly expressed genes. As you can see between these replicates, the number of genes detected is extremely consistent, making QuantSeq-Pool a reliable option for gene expression profiling. And compared to standard QuantSeq, there is a high correlation between the two methods when looking at gene expression. The slight differences noted here are mainly due to the low input that was utilized for QuantSeq, and this can actually be traced back to mitochondrial transcripts. A huge benefit to using QuantSeq-Pool is scalability. For example, if you have a total of 768 samples, you will need 8 96-well plates. After performing first strand synthesis, each of these 96-well plates would be pooled into one tube, leaving you with 8 reactions compared to 768 reactions. This substantial decrease in tubes improves consistency and reduces the stress of having to handle such a large number of samples at a time. For large-scale projects, QuantSeq-Pool can be scaled with the use of our UDI plates. Pictured here are 36,864 samples being processed in only 384 tubes with the use of 4 UDI plates, and can then be sequenced in the same lane. Other major benefits of QuantSeq-Pool is the time and cost savings. With conventional RNA-Seq, a significant amount of time is spent on QCing individual libraries. Additionally, this is a consumable cost, having to process each library independently. With QuantSeq-Pool, you're reducing your handling time for library generation and amplification due to early pooling, and you're also eliminating time spent on QCing and preparing equimolar ratios of your libraries prior to sequencing. But how do you know which kit is right for you, QuantSeq or QuantSeq-Pool? So here's a nice figure walking you through how to choose the appropriate kit. And this figure is also available on our website. If you're working with between 10 to 120 nanograms of RNA that is of medium to high quality and fairly homogeneous across sample types, QuantSeq-Pool would be the right choice for you. If you're working with lower or higher inputs that are of lower quality and diverse, we would recommend you to use standard QuantSeq. If you have any questions about deciding which kit is right for you based on your sample type, please reach out to us and we're more than happy to help. So with that, I would briefly like to recap the benefits of QuantSeq prior to beginning our question and answer section. Like QuantSeq, QuantSeq-Pool is a cost-efficient option for expression profiling, especially for large-scale projects that require a high degree of multiplexing. The workflow for QuantSeq-Pool is accelerated by early pooling of samples following reverse transcription and allows the researchers to go from 96 reactions just down to one tube. Depending on the needs of researcher, QuantSeq-Pool is scalable and you can multiplex up to 36,864 samples. Finally, UMIs are seamlessly included in the workflow and allow you to identify and eliminate any amplification bias. So with that, I want to thank you all for your attention.